Hi, this is Aya from the Chipster team. In this tutorial I will show you how to exclude samples from a dataset. So, for example, when you detect an outlier and you want to leave it out of the analysis, or when you have a multi-group experiment and you only want to analyze a subset, say compared to groups. So, as example data here I use RNA-seq data, but the instructions are the same for microarray data as well. So in my example session here I have two files, the actual data file which has expression values for different genes in different samples, and then I have the phenodata file which describes the experimental setup. So I make it a bit bigger so that you see it better. In this experiment cells were taken from 3 different patients with parathyroid cancer. The cells were cultured and then treated different ways, either with nothing or with DPN or OHT. So effectively we have 3 experimental groups and each group has 3 biological replicates. Now let's say that we want to compare only the control samples and the DPN treated samples, so we want to leave the OHT treated ones out. In order to do that, we make a new column. You can name it what you like. I will call it keep. And now in this column I will mark with 0 those samples that I want to leave out, so the OHT samples, and all the other ones I mark with 1, so the ones to be kept. Okay, so once that is done, I will select the actual data file, and then under the NGS category we have under the NGS tab there is the tool category utilities, and there you find the tool extract samples from table. If you are working with microarray data, exactly the same tool is available under the utilities category there. So now let's go back to the NGS and utilities and the tool, and before running the tool we need to set the parameters. Here specifically we need to indicate the phenodata column that contains those 0s and 1s, indicating which samples to get rid of. So that's the keep column in our case. And then I hit the run button. This will produce 2 files. So they will be, here they are, so they will be the actual data file, which is similar to the one before, except that now it only has 6 samples instead of 9. And then if we look at the phenodata file, you can see that it contains only those samples which we marked with 1. So this is how we exclude samples. That's all there is to it. I hope you found this tutorial useful.